The bigger picture of respiration, breathing in and out. We all do it. It's super important, right? We take in oxygen, we breathe out carbon dioxide. But is that all there is to respiration? Nope. Not by a long shot, science friends. That breathing, the in and out, that's just the start of an amazing journey. It's like opening the door to a giant, bustling factory. A factory that's working hard inside every single one of your tiny cells. It's science in action, happening every second of every day, even when you're fast asleep dreaming about, well, science. Think about it. When you breathe, you're getting oxygen. But why do your cells need that oxygen? It's not just to fill up your lungs like balloons, though that's part of the mechanics. That oxygen has a very special job to do, a crucial role in a much grander scheme. It's a key ingredient in a much deeper, much more incredible process. So, what exactly is this cellular respiration we're talking about? Let's get down to the nitty-gritty. Imagine your cells are like tiny, super efficient factories bustling with activity. These factories need fuel to run, just like a car needs gasoline or an electric car needs, well, electricity. The fuel for your cells comes from the food you eat, especially sugars like glucose. Cellular respiration is the amazing chemical process that takes this glucose and, with the help of that oxygen you just breathed in, breaks it down. It breaks it down to release energy, pure usable energy for everything you do. This energy isn't like the energy from a lightning bolt, zap. That would be way too much for a tiny cell to handle, like trying to power your phone with a power plant. Instead, the cell captures this energy in special little packets, like tiny molecular batteries. These batteries are called ATP molecules. ATP stands for adenosine triphosphate, which is a bit of a mouthful, but you can just call it ATP. It's the main energy currency of the cell, the dollar bill of the molecular world. Alright, science explorers, let's dive into the first big step of cellular respiration. It's called glycolysis. Say it with me, glycolysis. It literally means sugar splitting, and that's exactly what happens. This initial stage takes place in the cytoplasm of the cell, which is the jelly-like stuff outside the mitochondria. It doesn't even need oxygen to get started. So, it's a process that many different types of organisms can do, even those that don't breathe oxygen like we do. Pretty cool, huh? It's the universal first step for energy. So, what's the main player here? It's glucose, that simple sugar we get from carbohydrates in our food. Glucose is a 6-carbon molecule, kind of like a little chain with 6 links. In glycolysis, this 6-carbon glucose molecule gets broken down, or split, into 2 smaller molecules. These smaller molecules each have 3 carbons and they're called pyruvate. Think of it like taking a 6-piece chocolate bar and breaking it in half, giving you 2 3-piece sections. This splitting process isn't free. It actually needs a little bit of energy to get started, like an investment. After glycolysis has done its job of splitting glucose into 2 pyruvate molecules, what happens next? Well, if oxygen is available, and for us humans, it usually is, these pyruvate molecules get ready for the next big phase. But they can't just waltz into the mitochondria's main event, the Krebs cycle as they are. They need a little bit of modification first. This preparation step is often called the link reaction, or sometimes pyruvate decarboxylation. Think of it as the security checkpoint and dressing room before the main concert. The link reaction acts as a crucial bridge. It connects glycolysis, which happened out in the cytoplasm, to the Krebs cycle, which takes place deep inside the mitochondria. Each of those 3-carbon pyruvate molecules we got from glycolysis now moves into the mitochondrion. Once inside, a very important transformation occurs. First, 1 carbon atom is snipped off from each pyruvate molecule. This carbon atom combines with oxygen and is released as carbon dioxide. Yes, that's some of the CO2 you breathe out. What a journey for a carbon atom. With 1 carbon gone, the remaining 2-carbon molecule is called an acetyl group. Section 5, the Krebs cycle, spinning for power. Now we're heading into the heart of aerobic respiration, the Krebs cycle. You might also hear it called the citric acid cycle. Why? Because citric acid is one of the first molecules formed in this amazing cycle. This incredible series of chemical reactions takes place in the matrix of the mitochondria. That's the very inner compartment. The acetyl-CoA molecules we just made in the link reaction, this is their destination. They are about to be put to work to generate even more energy carriers. It's like a spinning wheel that churns out power. So, how does this cycle work? Well, each 2-carbon acetyl-CoA molecule enters the cycle by combining with a 4-carbon molecule that's already there. oxaloacetate. This creates a 6-carbon molecule. Hey, that's citric acid. Then, through a series of 8 carefully controlled steps, this citric acid molecule is gradually broken down and rearranged. Along the way, carbon atoms are stripped off and released as carbon dioxide. Yes, more of the CO2 you exhale comes from here. It's a bit like a molecular recycling plant, constantly reforming and processing. The main purpose of the Krebs cycle isn't to make a huge amount of ATP directly. In fact, for each turn of the cycle, and remember there are 2 turns for every glucose molecule because we got 2 acetyl-CoAs, only 1 ATP molecule is made directly. That's still useful, of course. But the real treasure produced by the Krebs cycle is the vast number of high-energy electrons it harvests. Section 6. The electron transport chain, the ultimate energy relay race. We've broken down glucose, we've made some ATP directly, and we've loaded up our electron carriers, NADH and FAD2. Now, it's time for the main event in ATP production, the electron transport chain, or ETC for short. This is where the vast majority of ATP will be made. The ETC is like an incredible microscopic relay race for electrons, and it happens in the inner membrane of the mitochondria. This membrane is all folded up, creating lots of surface area for this process to occur. It's a marvel of cellular engineering. So what happens here? Our energy-rich molecules, NADH and FAD2, arrive at the inner mitochondrial membrane. They drop off their high-energy electrons to a series of special protein complexes embedded in the membrane. These proteins are the runners in our relay race. As the electrons are passed from one protein complex to the next, a little bit like a hot potato, they lose a bit of energy at each step. It's a controlled release of energy, not all at once. This is super important for capturing that energy efficiently. Section 7. Chemiosmosis. Making ATP molecules galore. We've reached the grand finale of aerobic respiration, the part where all that stored energy in the proton gradient gets converted into mountains of ATP. This amazing process is called chemiosmosis. Say it with me. Chemiosmosis. It sounds complicated, but the idea is actually quite elegant. Remember that electrochemical gradient we just built up with the electron transport chain? All those protons packed into the intermembrane space of the mitochondrion are just itching to get back into the matrix, to where there are fewer of them. They want to flow back down their concentration gradient. Now these protons can't just diffuse freely back across the inner mitochondrial membrane. That membrane is pretty selective about what it lets through, but there's a special channel, a molecular machine, just for them. This incredible enzyme is called ATP synthase. Think of it like a tiny, super-efficient water wheel or a turbine. As the protons rush back into the mitochondrial matrix through this ATP synthase channel, they make the enzyme spin. It's like water rushing through a dam's turbine, making it turn and generate electricity. Section 8. Running on fumes. What about anaerobic respiration? So far we've been talking about aerobic respiration, the kind that uses oxygen. It's super efficient and makes a lot of ATP. But what happens if oxygen isn't around? Or what if an organism, like some bacteria or yeast, lives in an environment completely without oxygen? Do they just run out of energy and stop? Not necessarily. Many organisms, and even our own muscle cells sometimes, can switch to a different process called anaerobic respiration, or fermentation. Anaerobic means without oxygen. It's like running your energy factory on an emergency backup generator. Anaerobic respiration still starts with glycolysis, that initial splitting of glucose into 2 pyruvate molecules in the cytoplasm. So it still produces that small net gain of 2 ATP molecules per glucose. That's not much compared to aerobic respiration, but hey, it's better than nothing, especially in a pinch. The problem is glycolysis also produces NADH. In aerobic respiration, NADH takes its electrons to the electron transport chain, and NAD+ is regenerated, allowing glycolysis to continue. But without oxygen, the ETC can't run. So, NADH would build up and NAD+ would run out, stopping glycolysis. This is where fermentation pathways come in. Section 9, the final tally, ATP and why our cells rock. So, after all these amazing steps, glycolysis, the link reaction, the Krebs cycle, the electron transport chain, and chemiosmosis, how much ATP do we actually get from one single molecule of glucose when oxygen is plentiful? Let's do a quick recap of the energy harvest. Glycolysis gives us a net of 2 ATP directly. The Krebs cycle also chips in with 2 ATP directly. These are made by what's called substrate-level phosphorylation. It's like finding a couple of coins on the street. Nice, but not the big jackpot. The real treasure, as we've seen, comes from all those NADH and FAD2 molecules we've been collecting. These electron carriers ferry their high-energy electrons to the electron transport chain. As electrons move down the chain and protons are pumped, the ATP synthase enzyme goes to work during chemiosmosis. Each NADH molecule typically leads to the production of about 2.5 ATPs, and each FADH2 molecule yields about 1.5 ATPs. These numbers can vary slightly depending on the cell type and conditions, but they are good estimates for our energy accounting.